Welcome to the third of 3 presentations designed to help you prepare for the biology MTEL. Today we'll be looking at the open response questions, look at how they're kind of structured and some strategies on how to approach them, what you can expect. And then we'll also go through 20 more sample multiple choice questions. So the open response questions are going to give you some sort of a prompt. And that prompt is usually followed by some bullet points to give you additional guidance on what the expectations are of how you should respond. You're given an opportunity to either type or handwrite your response. I would probably type my response, but then add kind of hand-drawn illustrations or graphs or kind of figures where needed. The topical areas usually break down into a question on kind of molecules, inheritance and evolution, and then also one on organisms or ecosystems. You'll be evaluated on these areas here. So purpose, did you meet the expectations of the question as they were outlined, and maybe even bullet pointed, right? So as you're kind of creating your response, make sure you're referring back to the open response question, to that prompt, and going in and anywhere there's an opportunity for you to respond or something that it looks like they're looking for, make sure that you hit that so that as they're being evaluated, right, that evaluation will kind of get that checkbox checked. They'll be looking for the accuracy of your response, right? So if you're dealing with like the central dogma, right, or transcription or translation, how accurate are you with your use of some of these concepts or your description of some of these concepts and use of terms? Can you support your response with real examples? So that's a way to kind of build out and demonstrate your understanding. And then rationale, as a total product, has your response met the kind of the expectations of the open response question? Oftentimes it'll deal with some sort of a biological phenomenon. So this is a way I kind of interpret what the evaluation is looking for. And then you'll have to create, right, or modify or discuss a relative classroom activity. So did you do that is going to be, the purpose. And then on the bottom, the rationale is like, how well do all of those pieces kind of align with each other? So did your knowledge and understanding of that biological phenomenon really align well with the activity, right? Are the students that are going to engage with that activity be working towards an understanding of that phenomenon? Subject matter knowledge, again, that's kind of just how accurate and well-versed you are in that material, right? And then how well can you support it with examples and additional details. So as a strategy, right, make sure you're going back to that open response question and looking for, another way to think about this is don't give them an opportunity to take away points, right? If you know something and you're confident about it, get it in there. If there's a bullet point that's seeking some sort of a detail or aspect, make sure that you've checked it off so they can check it off. Make sure that you're really careful and intentful on how you're responding to these open response questions. You can lean a little bit on the prompt language and the way they're describing things to make sure that you're kind of talking using the same language, right? But avoid just appearing to have totally dependent on the language they're using. If you're looking for the types of details that you can kind of get in there, pause and think about what some sort of a phenomenon is, right? When and where it might occur. So if this is a cellular process, for example, let's say, just to go back to the central dogma, if we're talking about transcription and translation, right? When are these different processes occurring? Where are these different processes occurring, right? In the nucleus and the cytosol, right? These are the additional details that you can add in there if you're kind of struggling to think about what details to add. For experiments, right? use every opportunity to integrate lab safety in there, even if it's just to say that, that the school or institution's established policies will be employed, or if there's specifics that you can also integrate in there. If your example allows for that, then for sure you want to do that. And even if you're not doing any sort of wet bench experiment or even experiment, you could even say that because the exercise is just using laptops or hand-drawn materials, that no additional lab safety policies were required. The activities you should describe should be kind of creative and interesting, right? So you certainly want to avoid the suggestion that you're just going to go in and lecture to students about a phenomenon and then test them on it, right? So think about different types of activities that they could do that are more engaging and interesting, like creating a model, designing their own kind of maybe pamphlets about a topical area. Maybe you want to have students draw their understanding of a biological structure or process before your activity and then after, and see how well their own comprehension has improved. What of your own learning goals, right, have been met? So think about the idea that all students, and we know this, right? All people, all students are diverse learners. So to the extent that you can use diversity within your activity, right, it's going to benefit more of the students. Make clear connections between activity and the phenomenon, right? So this goes back to that kind of rubric table in terms of the rationale, or that it makes sense with your approach, lines up with your purpose. And then you can always think about an activity of what happens if things go wrong, right? Oftentimes things don't work the way that you expect. That's, a great thing to talk about within your open response. What happens when things go well? How would you expect the results or the outcomes of your activity to look, right? And what happens if they don't? Let's move on to the sample questions. First one, which of the following correctly matches a phylum with the most common mode of excretion by organisms in the phylum? So the correct answer here is D. These are all modes of excretion, right? But the only one aligned with the correct phylum, right, is Annelida with Metanephridia. Number 2, which of the following best explains why the body makes a more rapid and stronger response to an infecting pathogen after vaccination against the pathogen? Memory lymphocytes made in response to the vaccination are activated. Preformed antibodies made in response to the vaccination are stored in the spleen. The number of lymph nodes in the body increases in response to the vaccination. The material used for the vaccination is stored in the lymph nodes and spleen until needed during an infection. The correct response here is A. Memory lymphocytes made in response to the vaccination are activated. The other 3 responses don't make any sense. Preformed antibodies made in response to the vaccination are stored in the spleen. Antibodies could certainly be made in response to a vaccination, and then the titer will inevitably kind of decrease over time. I don't know anything about storing antibodies in the spleen. The lymph nodes are anatomical structures. They wouldn't change in number in response to a vaccination. They're kind of fixed in number and position. And the material used for the vaccination is stored in the lymph nodes and spleen until needed during an infection. That's also not true. So with the memory lymphocytes, what you see are after a response to an antigen, that could perhaps be a vaccination, you're going to get a primary response, or this could be a natural infection, right? So here you have lymphocytes are responding. You have B lymphocytes are a type of lymphocyte that makes antibodies, T lymphocytes are a class of lymphocytes that respond in other ways to help address an infection. But both of these, the B cells and the T cells, some of them will get to work to respond to the antigen or the vaccination or the pathogen. But some of them will also differentiate into memory cells, and they'll kind of slink off into the shadows and wait should there be a subsequent infection. And if there is, right, then the second response tends to be more robust. It tends to be quicker because they're already available and ready to go. They don't need to go through some of the initial sites or the initial stages of activation. So you have these memory cells that are a good match for the antigen or the vaccination or the infection, and they're primed and ready to go. So their response is quicker and tends to be more robust and more specific. So it's just a much more quality type of response. Which of the following plant structures most likely contains sclerenchyma cells? The primary phloem of a sunflower plant, the corolla of a hibiscus flower, the apical meristem within the terminal bud of a birch tree branch, or the pericarp of a hazelnut. So the answer here would be D. So the sclerenchyma, when I think sclerenchyma, I think like tough and rigid. So these structures tend to have cell walls that are rich in lignin, have a lot of integrity. So they would be found in more rigid, woody in nature types of plant tissues. So that would align with D. Here's a figure if you want to spend more time looking at a review of some of these different plant anatomy structures. This is a leaf with to the left would be on the bottom of the leaf and to the right would be the top of the leaf. I put this in here also because the phloem and xylem are highlighted as structures. And those are really common to encounter with MTEL types of questions, those different structures. So I would be sure to be comfortable with those. A scientist examining fossils of small rodents, found fossils of one species in a deep stratum and found fossils of a similar but distinct, more recent species in the stratum immediately above. Fossils of the transitional organisms were not found. Of the following, which is the best explanation of the observed distribution of fossils in the strata? So the answer here is A. One explanation for this would be a brief and rapid time of evolutionary change within that lineage. So few fossils of a transitional organism would be present. Which of the following best describes the difference between natural selection and artificial selection? Well, by definition, the correct response here is A, right? Environmental conditions drive natural selection, whereas humans determine artificial selection. Transmissible spongiform encephalopathies, TSE, are a family of neurodegenerative disorders caused by prions. Prions are infectious misfolded protein sequences that can enter cells and alter the folding of normal cellular prion proteins. A student claims that prions are a living organism because they are found in cells, they can reproduce, right, and respond to the environment. Which of the following statements about prions best refutes the student's claims? The best response here would be B, because one of the hallmarks of life, right, is that there's a demonstration of metabolic processes. So prions have no cellular machinery with which to carry out metabolism. All right, which of the following marine animals exhibits strictly bilateral symmetry? Right, C would be the right response here. This decapod, right, looks like a lobster. So this clearly has bilateral symmetry, right? A line down the midline would give something roughly as a reflection on the left that looks like a mirror image to that on the right. We have A and B with radial symmetry, right? D looks like a coral with no real symmetry. So C is the one here with bilateral symmetry. So question 8, which of the following best supports the endosymbiotic theory? So endosymbiotic theory doesn't have anything to do with embryonic development or organisms living within other organisms. So A and B are out. D doesn't make any sense with, I mean, that certainly happens, but it has nothing to do with endosymbiotic theory. The correct answer is C. So endosymbiotic theory seeks to explain the origins of double-membraned organelles like mitochondria and chloroplasts, and also looks to other evidence that they originated as independently living prokaryotic cells because they have DNA. So they have their own kind of mini organelle genomes, as well as these double membranes support the idea that they were historically independent prokaryotic cells. Number 9, a male mouse with black fur and short tail mates with a female mouse that has brown fur and a long tail. The offspring includes pups that resemble the parents, as well as some that are brown with short tails and black with long tails. The appearance of the pups of these two phenotypes that differ from the parental phenotypes is best explained by which of the following? So the correct answer here is B, the principle of independent assortment. So we can have genes, right, that are responsible for these different phenotypes, and if these genes are present on different chromosomes, these chromosomes can segregate independent of each other into different gametes, right? So in this way, you can get offspring that don't resemble the parents relative to these two particular traits. So we can see this by looking at an illustration of meiosis, right? So here we're starting with two different genes, and we can see that they get initially duplicated. These two different chromosomes, right, get duplicated, and then through meiosis 1, the duplicated chromosomes segregate into distinct daughter cells, right? And then through meiosis 2, you can see that the haploid kind of product of meiosis gives us the variability in the different types of chromosomes that each of these different gametes is carrying. Right? So all of these different chromosomes segregated independent of each other. If they were giving rise to the kind of fur color and tail length in mice, then they can be independently kind of segregated into gametes, and they behave genetically kind of independent of each other. All right, which of the following is the sexually mature diploid structure of flowering plants? And the answer here is the sporophyte. Right, so shown here with this cycle, looking at the gametophyte and sporophyte stages here, right, we can see that the sporophytes are the diploid, right? So that's denoted with 2N. The difference in which of the following is most likely to be a prezygotic barrier to successful hybridization between two species? So mitochondrial DNA is not going to influence the ability for zygotes to to fuse or for one to fertilize the other, right? Heterozygosity and length of gestation, neither of those are going to play a role. Acrosomal proteins, for sure. So biochemically, there could be an incompatibility with the ability of a sperm to fertilize an oocyte. So D would be the correct response here. So there's various different prezygotic kind of barriers or isolation mechanisms. It could be because two individuals, are unable to form a zygote because their gametes never interact because they're somehow isolated in different habitats. Or maybe something about the behavior of mating or the opportunities for parents to interact, right, is somehow inhibited by time. So there could be behavioral mechanisms at play that don't allow things to align the way they need to. Mechanical isolation potentially in terms of anatomy, right? Or gametic isolation, right? Which I think is what the acrosomal kind of biochemical compatibility is referring to. Of the following, which is most likely defective in a person who is not getting enough oxygen delivered to his or her tissues? So these are all tied to blood, right? But only one of these is linked to the oxygen carrying capacity of blood, right? So that would be hemoglobin, right? Platelets, those are for clotting, right? Plasma is the liquid portion of blood, right? And white blood cells are the leukocytes that are important for immune function. So the correct response here would be hemoglobin. All right. Biopsies of malignant tumors often include a karyotype, and the results sometimes reveal a chromosomal translocation. Which of the following best explains why translocations can cause cancer? So the best response here is B, because when you get translocations, right, that's movements of one part of a chromosome to another, right, and those can be identified through techniques using a karyotype. A karyotype is where we kind of arrange the chromosomes, and they can identify chromosomes based on size and banding patterns, but also with more sophisticated techniques can be used to identify based on signature genes that are on each of these chromosomes. You can see where some chromosomes have exchanged material with each other, which is not typical. Right, so with these translocations, sometimes you can end up with a region of a DNA that now there's an introduced flanking of one part of DNA next to a regulatory sequence of another gene on another part of a chromosome. Right, so that can start to influence the way genes are expressed. So that is the type of thing that can be associated with cancer. These other responses are either misleading, misguided, or don't really make any sense. So the right answer here is B. Which of the following is most responsible for the movement of water up the stems of tall plants? So the right answer here would be A, the hydrogen bonding between water molecules, which that goes way back to our first presentation looking at some of the unique properties of water, and one of them is this hydrogen bonding, which has the intramolecular kind of attraction between the molecules. In humans, which of the following combinations of a stimulus and a hormone released in response to the stimulus interact in a positive feedback loop? So the correct answer here is C. So looking at the other responses, they deal more with negative feedback loops, right? But a positive feedback loop would be an example of oxytocin, which in several different circumstances, the release of the hormone oxytocin from the posterior pituitary actually through its release or the action that it has downstream causes the release of more oxytocin. So that's one case here with breastfeeding, where the suckling of the infant can induce the expression of breast milk, but then also the release of more oxytocin, and that can lead to a positive feedback loop. A plant breeder cross-pollinates a true breeding plant with red flowers with another true breeding plant of the same species that has white flowers. All of the F1 plants have pink flowers. When any F1 plant is cross-pollinated with another F1 plant, individual F2 plants that are produced have red, white, or pink flowers. The data best support which of the following inheritance patterns for flower color in this species of plant. So we've touched upon a couple of these throughout the presentations, right? But this is an example of incomplete dominance, where a heterozygous individual for this particular trait, right, is going to have pink flowers, right? But then if you cross those individuals, right, you'll see again either the dominant recessive or the heterozygous intermediate kind of traits manifest and express themselves. As levels of carbon dioxide rise in Earth's atmosphere, which of the following is the most likely consequence? The correct response here is A, trapping by greenhouse gases of the heat energy emitted by the Earth's surfaces. Trapping by greenhouse gases of the heat energy emitted by the Earth's surface will increase, contributing to global warming. All right, nuclear envelope breakdown and chromosome attachment to spindle fibers is completed during which of the following stages? Right, so this is A, prometaphase. Right, so this is late prophase, where you get a disassembly of the nuclear envelope and then a connecting of the microtubules of the mitotic spindle to the centromeres of the chromosomes. Some vascular plants produce defensive chemicals such as nicotine and tannins that deter herbivores. These chemicals are primarily stored in which of the following compartments of plant cells? The correct response here is the vacuoles, right? So these are the organelle structures that are present in plant cells that you don't see in animal cells. They can act as kind of storage vessels, as well as help to osmoregulate and osmoregulate, so they help to balance the movement of fluids and electrolytes within the cell. This is a beautiful histology image showing plant cells, right? So here you can see chloroplasts and cell walls, right? But here's also vacuoles within those structures. Right, a diploid cell that produces haploid products by going through 2 consecutive rounds of nuclear division is best described as which of the following? Right, so this is a germ cell. Right? So mitosis entails DNA replication and then cell division, DNA replication, then cell division. And then meiosis is cell division and then another round of cell division. Right? So you get DNA replication, division. So this you only see, right, in animals, you only see this in the germline. So this is the only time you would see meiosis would be in a germ cell. Okay. So today we looked at the open response question format and some strategies for answering them, what to expect. The DESE MTEL site has some great examples of these open response questions, as well as the way they were kind of scored. So it presents a couple kind of weak responses, a couple strong responses, and then looks at how those different types of responses were assessed. And then we also spent some time going back through some multiple choice questions. So again, the intent here is to give you some experience in the types of questions and what the questions might look like when you encounter them on the MTEL, but also to flag any areas that you think you could use more practice on in terms of preparing. Hey, thank you. Good luck on the MTEL.